Welcome to the presentation of our online course, Digital Scale-Up and Optimization of Animal Cell Culture Bioreactors. Through our online courses series, you learn the principles of bioprocess engineering and their applications to enzymatic bioconversions, microbial fermentations, and animal cell cultures processes. For more details, visit us at bioprocessengineeringonline.com. Animal cell culture is a key technology in the production of biopharmaceuticals. Most modern pharmaceutical manufacturing uses animal cells to produce recombinant glycosylated proteins, especially monoclonal antibodies, viruses for vaccines and gene therapy, and active cells for use in cellular therapies. When developing new animal cell culture processes for biopharmaceutical production, bioprocess engineers must scale up cell culture bioreactors, starting from small-scale systems like roller bottles and spinner flasks used for initial cell propagation, and progressing to large-scale stirred tank bioreactors capable of producing kilograms of therapeutic products. A major challenge for bioprocess engineers is to precisely tailor bioreactor operation to the specific cellular biotransformation of interest. Key factors in bioreactor design and operation include selecting the appropriate operating mode, batch, fed batch, or perfusion, optimizing critical parameters such as substrates concentrations, feeding flow rates, product induction timing, and total culture duration. Careful control of these elements is essential to maximize productivity and ensure process robustness. To design high-performance cell culture bioreactors, engineers rely on the advances of bioprocess engineering. This scientific discipline provides a conceptual and methodological framework for the simulation, scale-up, and optimization of animal cell culture bioreactors. Its ultimate goal is to enable the most efficient and productive use of animal cells in biomanufacturing processes. To accelerate process development and scale-up, bioprocess engineering increasingly relies on knowledge-driven simulation models built on a deep understanding of key physicochemical and biological phenomena. These models act as digital twins, virtual representations of bioreactors, enabling in silico experimentation across a wide range of operating conditions. Typically developed using laboratory-scale experimental data, these models play a crucial role in optimizing operating modes and process parameters, ultimately supporting goals such as maximizing productivity or minimizing production costs. By enrolling in this course, you will develop a solid foundation in the digital methodologies of bioprocess engineering with a focus on the scale-up and optimization of cell cultures bioreactors. The course is structured in two key phases. One, model development from laboratory data. You will learn how to apply core bioprocess engineering principles to develop mechanistic simulation models based on laboratory-scale experiments. These models will be tailored to the specific cellular strain used, enabling you to capture and describe the key phenomena influencing cellular growth, substrate utilization, and product formation. 2. Simulation-guided scale-up and optimization. Building on your model, you will explore how to use simulation tools to scale up cellular processes and optimize operational strategies. Emphasis will be placed on selecting suitable bioreactor technologies and operating modes, batch, fed batch, or perfusion, to enhance the efficiency of cellular biocatalysts and maximize overall process productivity. Throughout the course, the bioprocess engineering methodology will be illustrated through a case study on the production of a monoclonal antibody using an engineered Chinese hamster ovary CHO suspension cell line. The production process is scaled up in a stirred and aerated bioreactor. Following an initial growth phase at 37 degrees Celsius, the temperature is lowered to 32 degrees Celsius to slow down cell proliferation and enhance monoclonal antibody production. The course is structured into two parts comprising a total of 7 sessions. Part 1, Understanding and Simulating the Cell Culture, contains 4 sessions. Principles of Cell Culture Engineering, Kinetic Analysis of Culture, Oxygen Transfer and Consumption in Bioreactors, Simulation Model Development. Part 2, Scaling Up and Optimizing Bioreactors, is made of 3 sessions: Batch Culture Optimization, Fed Batch Culture Optimization, Perfusion Culture Optimization. The 7 sessions can be taken independently. In the first part of the course, you will develop a knowledge-driven simulation model of the cell culture process. This model is built on a scientific understanding of the key phenomena that govern cellular metabolic activity in bioreactors, including substrate consumption, glucose, glutamine, and oxygen, cell growth and death dynamics, production of growth-associated metabolites, lactate, ammonia and carbon dioxide, and synthesis of the monoclonal antibodies. This mechanistic approach allows you to simulate and predict cell behavior under varying conditions. Session 1, Introduction to Bioprocess Engineering Principles, is designed for learners new to bioprocess engineering. The session provides a solid foundation in the essential principles needed to understand and model cell culture processes. You will be introduced to 3 core topics: kinetics of animal cell cultures, mass transfer in bioreactors, and mass balances applied to bioreactor systems. The first step in developing a simulation model is to analyze the kinetics of the specific cell culture. In Session 2, you will learn how to conduct a kinetic analysis by combining data from both batch and continuous bioreactor experiments. In batch cultures, you will examine cell behavior under initially high concentrations of glucose and glutamine to characterize growth and substrate consumption dynamics. In continuous cultures, you will explore how nutrient limitations, glucose and glutamine, affect cellular metabolism, enabling the identification of key metabolic shifts under stress conditions. This dual approach provides a comprehensive understanding of how cells respond across a range of substrate concentrations, critical for building accurate simulation models. In both batch and continuous cultures, you will measure the time-dependent variations of key components in the cell culture, including substrates, glucose, glutamine, and oxygen, viable and dead cells, metabolic byproducts, lactate and ammonia, and the produced monoclonal antibodies. In Session 2, you will further explore cell culture kinetics by determining the key reaction rates that govern the process. Using experimental data, you will sequentially calculate culture rates of cell growth, death, and metabolism, and the corresponding specific cellular rates. Understanding how these rates evolve over time is essential for developing accurate and predictive simulation models of cell culture behavior. In Session 3, you will explore the continuous aeration process in cell culture. You will analyze the kinetics of oxygen transfer, solubilization, and consumption within the bioreactor, as these are critical factors that often limit culture performance. You will also identify the rate law for oxygen consumption by cells, the thermodynamic law governing oxygen solubility in the medium, and the kinetic law of oxygen transfer from the air phase to the liquid medium. In Session 4, you will learn how to build a simulation model based on the insights gained from the 2 previous sessions. You will begin by identifying the fundamental phenomena that influence cell culture kinetics, including cellular reactions, oxygen and carbon dioxide solubilization and transfer, which are influenced by fluid flow within the bioreactor. Next, you will formulate the mathematical relationships that define the model. This involves combining thermodynamic and kinetic laws governing the core biological and physical processes, with mass balance equations specific to the bioreactor's operating mode. The key kinetic laws to be identified in the simulation model are those that describe cellular activities as a function of the culture medium composition. These include rate expressions for cell growth and death, glucose, glutamine, and oxygen consumption, lactate, ammonia, carbon dioxide, and antibodies production. Additionally, you will learn how to numerically integrate the differential and algebraic equations that govern the time-dependent changes in cells, substrates, and products concentrations using a spreadsheet. The second part of the course covers the digital scale-up and optimization of cell culture bioreactors. Using the previously developed simulation model, you will compare culture's kinetics across three operation modes: batch, fed batch, and continuous perfusion. For each mode, you will optimize key operational parameters to maximize bioreactor productivity. Productivity, measured in grams antibodies per liter of bioreactor per hour, serves as a key indicator of the bioreactor's utilization efficiency. In cell cultures, achieving maximum productivity or process intensity requires careful consideration of oxygen transfer limitations. The attainable cell concentration is constrained by the oxygen transfer rate. The highest cell density is reached when the rate of oxygen consumption in the medium equals the maximum rate of oxygen transfer from the gas phase. That is, when the dissolved oxygen concentration drops to its minimum allowable level. You will learn how to determine this maximum cell concentration limited by oxygen transfer in all three batch, fed batch, and perfusion modes. Session 5 is devoted to the digital scale-up and optimization of the batch culture. Batch culture, where the entire culture medium is introduced at the start, is the simplest and most straightforward culture mode. However, batch culture tends to yield the lowest viable cell concentrations. At high initial concentrations of glucose and glutamine, substantial amounts of lactate and ammonia are produced as byproducts. This accumulation leads to cell death, which significantly limits the maximum viable cell concentration that can be achieved. The main operational variables in batch culture for antibody production include the initial concentrations of glucose and glutamine, the timing of the temperature shift to 32 degrees, the total culture duration. Using the simulation model, you will perform virtual experiments to evaluate how these variables influence cell culture kinetics and the maximum attainable concentrations of cells and antibodies. Finally, you will identify the optimal set of operating conditions that maximize batch bioreactor productivity. Session 6 focuses on the scale-up and optimization of fed batch processes for antibody production. In fed batch culture, the bioreactor is initially partially filled, and fresh medium is gradually added throughout the process. The feed flow rate is dynamically adjusted to maintain low, near steady-state concentrations of glucose and glutamine. Maintaining limiting substrate concentrations helps to minimize the production of inhibitory metabolites such as lactate and ammonia, reduce cell death caused by byproducts accumulation. As a result, fed batch culture can achieve higher viable cell densities and greater productivity compared to traditional batch processes. Fed batch cultures are more complex to operate than batch systems due to the greater number of operational variables involved. Key variables include the initial volume fraction of medium in the bioreactor, the substrate concentrations in the feed medium, the target steady-state concentrations of glucose and glutamine, the timing of the temperature shift. Through virtual experimentation using the simulation model, you will evaluate how these variables influence culture kinetics and antibody production. You will then optimize the operational conditions to maximize bioreactor productivity in fed batch mode. In Session 7, you will explore the continuous perfusion mode of cell culture, a technology designed for achieving very high cell densities. In this system, cells are retained using an external microfiltration membrane module, while fresh medium is continuously perfused through the bioreactor. To support high cell densities, the air supply is enriched with oxygen to overcome potential oxygen limitations. Additionally, the process may include cell bleeding, where a small fraction of cells is regularly removed. This helps maintain a steady-state concentration of viable cells, optimizing long-term productivity. Optimizing perfusion culture involves several key operational variables, including the substrate's concentrations in the feed medium, the target steady-state concentrations of glucose and glutamine, the cell bleeding percentage, the time of temperature change. Through virtual experimentation, you will evaluate how these parameters influence cell growth and antibody production under continuous perfusion conditions. The session concludes with the identification of the optimal set of operating conditions to achieve maximum antibody productivity. The course resources include recorded slideshow presentations covering animal cells kinetics, bioreactor simulation models, oxygen transfer and consumption in bioreactors, cellular bioreactors operation and optimization. In the first sessions of the course, you will use spreadsheet templates with a green-red correction system. On spreadsheets, you will analyze cellular growth and metabolism kinetics and determine the oxygen transfer rates and coefficient. In the second part of the course, you will use spreadsheet simulators to explore the operations of batch, fed batch, and perfusion culture modes. The spreadsheets will allow you to visualize the kinetic impact of operational variables and identify the optimal culture conditions. You have the flexibility to tailor this online course to your individual pace. Plan and structure the course according to your own schedule. Additionally, coordinate the interactions with your tutor through video meetings. For further information on the course, please visit our Bioprocess Engineering online website or contact us at bioprocessdigital.com.